I'm going to spend a few minutes here talking to you as a journalist and a layperson, and I'm often the only one among scientists. But I actually jumped into self-testing as a journalist and a communicator trying to tell a story. So I'm going to just share with you a little bit, and some of you have seen aspects of the Experimental Man Project and the book I wrote about that. But I'm going to give you a just short little overview of that and then some of the newer stuff that I have done. But first, since we're called pioneers, I was thinking, DNA extraction through the ages. And it's a little known fact, actually, that the ancient Egyptians were extracting DNA. And this, in fact, comes from a tomb. And that is doing a buccal swab. From the ancient times. It was done in the Middle Ages. The Mayans did it. And you go back, and there are lots of different ways that people have been extracting blood and samples through the ages. And I only say that as a person, again, not a scientist, who's been poked and prodded and liters of blood taken. And this is the way we do it now. And I think I'm up to 6 or 7 liters at least. And I sort of lost track after a while. But thank God we finally invented the saliva test for DNA. Anyway, my journey started back in 2001, and this was just after the announcement of the rough draft of the Human Genome Project. And I was given the assignment at Wired magazine to explain this thing to Wired readers. And I'd been covering healthcare, actually, up to that point, and was beginning to get into something called biotechnology, which I was very excited about covering. But this is how I got into this. It was an assignment. We were sitting around going, how do we explain let's see, DNA, what is this? And so I got this crazy idea of why don't I get tested? And in fact, no one had ever been tested until we found out later that Craig Venter, no name person anyway, Craig Venter had tested himself, which we didn't find out until 2003. And the Human Genome Project itself was, of course, a compendium of several people. So this is me getting my initial sample in Sequenome in San Diego, and this was mass spectrometry at the time. There wasn't the sort of advanced sequencing we have now. That was an ancient robot that did my sequencing. This is how we listed it. There was no fancy 23andMe sites and all of this. It was just on a big spreadsheet. It was a very big one. In fact, I put it on a wall. It was many, many feet long, my first results. And then this was the original Wired story. And I've always been annoyed at what they called this story, which was DNA as destiny. And I said at the time, DNA is not really destiny. And I've been sort of dogged with that for many years. But when you turn into the story itself, 100% genetically analyzed, which, seeing as we're here talking about the PGP and the fact that complete genomes now are beginning to happen in a big way, this was pretty ridiculous because I had 400 SNPs done, not exactly 100% genetically. Anyway, we skipped to 2006, and the other big moment for me as a self-tester, again, as a journalist self-tester, was National Geographic called me up, an editor there, and said, you did your genetic testing, how about your environmental testing? So I went out and did this story called The Pollution Within, and I was tested for hundreds of environmental toxins, the levels inside of me. And this was actually the second most read article in 2006 in National Geographic, barely behind soccer, which I thought was kind of cool, that people thought we were almost as important as playing soccer. And what they did was, for this article, they had me go out and do, in great National Geographic fashion, a sort of autobiography of my chemical past. So where I grew up, that sort of thing. That was my house. This is a chemical house that we did. And this is still up if you want to go look at it, National Geographic. I got a chemical report card. These are all the various chemicals like dioxins, phthalates, et cetera, that I was tested for. And this is the beginning of the story. My journalist as guinea pig experiment is taking a disturbing turn. I'll let you read the rest of the story. And then this was the book that came out in 2009. Anyway, just to give you a brief overview of some of the high points of the Experimental Man Project. Thousands of tests, hundreds of labs. I had all kinds of people, including George, help me out with this. I'm up to 24,000 genotypes annotated, all these toxins, lots of brain scans and scanning technologies, my proteome, my microbiome. This is an honor roll of all the various companies that helped me out in labs. And I'll just give you a very quick overview of what some of the things that I found. Basically, I started the project healthy, which is important. And in fact, your exam ends at that point today. And then I went and had things like brain aneurysm. I don't have time to get into a lot of this here, but you can get the sense of the way it worked. I had my brother and I both sequenced, and this is where it gets a little serious with genetics. You know, a lot of the 24,000 genotypes, by the way, have not changed my life. I mean, I wish I could say they have at this point, but I think they will eventually. But this was a serious case with my brother who has osteogenesis imperfecta. And we did actually sequence both of our collagen genes and discovered what was going on with him. So this is a rare genetic disorder. My favorite genetic marker, however, remains, if you all have seen this talk before, the caffeine fast metabolizer gene. And in fact, I wish I had a cup of coffee up here right now. Environment, this is levels of environmental toxins inside of me, things like DDT, and these are all compared to Centers for Disease Control levels. And I don't have time to get into this, but you can check this out at experimentalman.com. Also, compared, since we have a gene environment meeting here, one of the things that I had a really interesting time doing in the book and in the project was actually taking my genotyping and seeing if I could find out anything about environmental exposure. And it turns out that there are lots and lots of genes and lots of data out there where one can find out your genetic sensitivity to different environmental factors. And this is on mercury. And in fact, my friend and colleague, Greg Stock, will be telling you a little bit about a company we founded on this called Ecoios later on. And just quickly here, I also did brain scans. I found out, thankfully, I have no trace of Alzheimer's. My greed level is normal. Tested for greed. My memory is a 25-year-old, and when I had that, I said, forget it, I'm not going to get tested again. Anyway, I'm going to stop there. I have a few more slides here, but I'm getting the high sign from Dan up here. But anyway, I think you all get the idea here. And the testing has continued. We're doing a lot of proteomic testing, microbiomic, and iPS stem cells. I've had an iPS induced pluripotent stem cell line done of myself, and we're about to do some really freakish experiments on that because they want to introduce some mutations into these cells and then see what happens when we start growing heart cells, brain cells, et cetera, from these induced pluripotent stem cells. But anyway, I'll stop there, and thank you all.